Today, I am taking up the topic replication of DNA. Replication is one of the important properties of the genetic material. Then, replication means making an exact copy of itself or making a replica of itself. You should not confuse between replication and transcription. Replication means DNA makes its own copy. Transcription means DNA makes one mRNA strand for participating in protein synthesis. Then coming to replication of DNA. It's a very complicated process and involves a number of enzymes. And in eukaryotes, replication takes place during cell division, during S phase of cell division. During other times, the DNA remains in a highly coiled state or in a supercoiled state. So before the transcription, the coiled DNA must uncoil, then only transcription is possible. Then, in DNA synthesis also, the three processes, initiation, elongation and termination are present, catalyzed by different types of enzymes. Then, DNA replication occurs in a semi-conservative fashion or in a semi-conservative way. That means, let us say this is the parental DNA. Parental DNA has two strands, this strand and this strand. One strand runs in 3-5 direction and another strand runs in 3-5 direction. Then, during replication, these two parental strands synthesize their own copies, which are known as daughter strands. So this is the synthesis, what happens. This is the original strand, parental strand. This is the daughter strand. It means during replication, the daughter DNA receives one parental strand and one new strand. So it is known as a semi-conservative way. And the proof for semi-conservative way comes from the experiment conducted by two scientists, Matthew Michelson and Franklin Stahl, in the year 1958. First I will describe how the replication takes place and then I explain this experiment. So replication always begins at a specific point known as origin of replication. Origin of replication. Before replication begins, as I have told you, the coiled DNA has to uncoil. Then, of course, here the enzyme topoisomerase helps in uncoiling the DNA. Next, the two strands of DNA are separated with the help of another enzyme that is known as DNA helicase. DNA helicase helps in separating the two parental strands by creating a replication fork. This is called replication fork. So, the two parental strands are separated from each other by the enzyme DNA helicase by creating a replication fork. Then, each strand, here, this one, they form templates. Templates means a pattern or a mold which helps in creating its counter copy. So, both the strands participate in replication. That's a very important point. In the case of transcription, only one strand participates. But in the case of replication, both the strands participate. Then, there is another important enzyme, DNA-dependent DNA polymerase, or simply DNA polymerase. The DNA polymerase enzymes are many types. DNA polymerase 1, 2, 3, 4, 5, 6 like that. Here, DNA polymerase 3 is very important. It alone participates in replication. The full form is DNA-dependent DNA polymerase. It can be simplified as DNA polymerase. You should not confuse this enzyme with RNA polymerase or DNA-dependent RNA polymerase. That is different. DNA-dependent RNA polymerase participates in transcription. But in replication, DNA-dependent DNA polymerase participates. And here it is the enzyme 3. Then, I have told you, both the parental strands participate in replication. But there is a complication. See this strand. This is running in 3-5 direction. This parental strand is running in 3-5 direction. DNA synthesis always occurs in 5-3 direction. Occurs in 5-3 direction. But the polarity of the parent strand is different. One is having 3-5 direction polarity, other one has 5-3. If you take from this side, 3-5, this is 5-3. This is a very important point. Then, how it happens? Let us take up this parental strand. This is in 3-5 direction. There is no problem as far as this strand is concerned, because DNA strand is synthesized in this direction, in a 5-3 direction. So this strand is anti-parallel to this strand, parental strand. The newly synthesized DNA strand is anti-parallel to the parental strand. So there is no problem. Transcription or replication of this strand takes place continuously from 5N to the 3N. It's a continuous synthesis. And this newly synthesized DNA strand is known as leading strand. Then coming to this strand. This is in 5-3 direction. 5-3 direction. But DNA synthesis can occur only in this direction. 5-3 direction. Then how it is possible? This problem was answered by a Japanese scientist known as Wakajaki. He suggested that on the strand, which is having 5-3 polarity, when the DNA strand or fork opens up to some distance, small segments are synthesized in 5-3 direction. So for this newly synthesized strand or fragment, we say it is synthesized from here. 1, 2, 3 like that. The nitrogen bases or nucleotides are added. So here the nucleotides are added continuously from one end to the other end. But here, from some distance, nucleotides are added in this direction, in 5-3 direction. These segments are about 150 to 200 bases long. And these fragments are known as Wakajaki fragments after the name of the discoverer. So, in this strand, Wakajaki segments are synthesized like this. First this fragment, then this fragment, then this fragment like that. They are synthesized continuously as the DNA unfolds or the double helix unfolds. These fragments are, I have told you, synthesized in 5-3 direction. Then these fragments are joined by the enzyme DNA ligase. It closes the gaps present between different Okazaki fragments. In this way, DNA replication occurs in this direction, 5-3 direction, and the newly synthesized daughter strand. So in this way, this problem is solved. Once replication is over, the process is terminated. The process is terminated. Any mistake done during this replication causes a mutation. But there is a proofreading mechanism. To prevent mutations in DNA, there is a proofreading mechanism. So mutations are very, very rare. Then here we have to remember one more point. Replication of DNA is a very costly process. It requires a lot of energy. So the nitrogen bases are the nucleotides. Nucleotide means it has a phosphate group, pentose sugar, and a nitrogen base. The nucleotides are in triphosphate form. These are all triphosphates. That means they have a lot of energy. For example, we take ATP. ATP means adenosine triphosphate. Adenosine is one of the nitrogen base. It occurs in triphosphate form. It has a lot of energy. Similarly, guanine occurs in the form of guanine triphosphate. Cytosine occurs in the form of cytosine triphosphate. Thymine occurs in the form of thymine triphosphate. In this way, these triphosphates are present in the nucleotides, and when the nucleotides are added to the OH end of the growing DNA polymer, the energy present in the nucleotides is utilized for joining or linking different nitrogen bases or the different nucleotides. So this is also a very important point. Then, how or what is the proof to say that DNA replication occurs in a semi-conservative way? The explanation is very good, but we also require a proof to show that DNA replication is actually is a conservative process. So I told you what is a conservative process. In a conservative process, what happens? The daughter DNAs have one parental strand and one new strand. So it's called semi-conservative method. Then, in DNA, nitrogen is present. And that nitrogen is 14N. 14N. Normally, the nitrogen present in DNA is 14N. Then these two scientists, Matthew Michelson and Franklin Stahl, used 15N in the place of 14N. 15N of nitrogen is a heavy nitrogen or heavy isotope of 14N. 15N is heavy isotope of 14N, but not radioactive. It doesn't emit any radiation. Only its weight is more. And that weight helps in identifying the different types of strands that will come to the point later on. So what they did, they have selected the bacteria E. coli or Escherichia coli. Generally, in the E. coli, the nitrogen present in the DNA is 14N. What they did, they have raised the E. coli on a medium, ammonium chloride medium, having 15N, not 14, 15N. So they have raised the E. coli on ammonium chloride having 15N for many generations. For many generations. As a result, what happened? 14N was replaced by 15N. So in both the strands of DNA, 15N is incorporated in the place of 14N. And 15N, DNA with 15N is heavy. Then, the time for replication of DNA in E. coli is about 20 minutes. It is about 20 minutes. But according to some people, it is more than 20 minutes. It is about 38 minutes according to some persons. But generally, let us take up 20 minutes. So, the duration of time taken for replication of DNA in E. coli is 20 minutes. So, when Michelson and Stahl raised E. coli on 15 nitrogen having ammonium chloride for many generations, in both the strands, 14N is replaced by 15N. This is heavy DNA. Then they have selected these E. coli and raised them on ammonium chloride having 14N. This is normal ammonium chloride or lighter ammonium chloride having 14N. So the time taken for replication is 20 minutes. After 20 minutes, they have isolated the DNA and studied. In this process, I have told you it is a semi-conservative process. The two daughter DNAs produced by the parent DNA must have one parental strand and one de novo, newly synthesized strand or a daughter strand. The parental strand inherited from the parent has 15N and the newly synthesized strand has 14N. It means the DNA is a hybrid one. First DNA is hybrid one. Then these two hybrid strands are allowed to multiply for another 20 minutes. That means after 40 minutes of raising E. coli on 14 NH4Cl, we get these DNAs. So this is the first generation. In the first generation, what do we get? Two hybrid DNA molecules. In the second generation, see, from this 14N, this is synthesized. In this DNA molecule, both the strands have only 14N. From this one, 15N strand, we obtain one hybrid DNA. Here also same thing. From this strand we get hybrid DNA and from this we get lighter DNA. So, in the second generation what we get? We get two lighter DNAs and two heavy hybrid DNAs. These are hybrid DNAs, these are lighter DNAs. If you allow multiplication for another 20 minutes, that means if these DNAs multiply, then we get this one. From this one we get two lighter DNAs. From this we get one lighter and one heavy DNA. One lighter and one hybrid DNA. From this we get two lighter DNAs, and from this we get one lighter and one hybrid DNA. This is very good to explain. But is it true? Yes, it is true. They have proved it. How they have proved it? After 20. So first they have taken some E. coli, raised on 15 NH4Cl, taken out their DNA and centrifuged in a solution called cesium chloride. They are centrifuged for about 20 hours. When they centrifuged the DNA from this E. coli raised on 15N, they got a sediment here like this, at this level. So the heavy DNA settles down here, forming a band-like structure. Then, after 20 minutes of replication, they have again subjected the DNA for centrifugation for 20 minutes, 20 hours. Then what happened? Instead of precipitating at this level, DNA precipitated at this level, the higher level. Here density is very important. If the DNA is heavy, the band forms at lower level. If the DNA is lighter, the band forms at higher level. Because it is heavy, this band is formed at lower level. But this one formed at a bit higher level than this band here, indicating the density of this DNA is less than the density of this one. This is heavy because it has 15N. It is lighter than this one because it is a hybrid DNA in which one strand is having 15N and other strand is having 14N. If the DNA is analyzed after 40 minutes, G1, here you find two lighter DNA molecules and two hybrid DNA molecules. So if you centrifuge after 40 minutes, you get bands like this. See the width of this band. This is two times to this one, this band. That means this is hybrid DNA. It is 2 times more than this one. But here, the DNA is separated into two bands. This band is mixed hybrid DNA, and this one is lighter DNA. Lighter DNA means these two strands. This is light DNA, this is hybrid DNA, this one. So both are equal. But the lighter DNA is formed slightly above the hybrid DNA. If you continue the multiplication further for another 20 minutes and we extract the DNA, after 60 minutes what happens? We get only 2 types of DNA, that is hybrid and light DNA in 3:1 ratio, in 1:3 ratio. See here, see? There are only 2 hybrid DNA molecules, but 2, 3, 5, 6 lighter DNA molecules. So there are 2 hybrid DNA molecules and 6 lighter DNA molecules. 2:6 means 1:3. See here. This is the result after 60 minutes. Hybrid DNA is formed, see, this one like this. This is lighter DNA. See the width of this band. This band is 3 times wider than this band. It clearly shows that lighter DNA is 3 times more than the hybrid DNA. So in this way, Michelson and Stahl have provided experimental proof in support of the semi-conservative nature of DNA. So they have supported the semi-conservative theory of DNA replication. And this proof is undisputable. This is undisputable. And the scientific world has accepted the explanation or the experimental proof provided by Michelson and Stahl. So in this way, DNA replication always occurs in a semi-conservative fashion. That means the daughter DNA always receives one strand from the parent and it synthesizes the second strand on its own, and it is known as daughter DNA. So that is about replication of DNA. In the next class, we will take up the transcription of DNA. So as usual, I want to give you one information. I have written these books. If anybody is interested, they can download them from Amazon. These are only kindly edition books. There is no print form, and each book costs about 500 rupees. They help you a lot in the ensuing 2024 NEET examination. Thank you.